continuación, disertará el Dr. Koenraad van Hoorde. Koenraad es doctor en bioquímica por la Facultad de Ciencias de la Universidad de Gante, jefe de servicios del Laboratorio de Patógenos Transmitidos por Alimentos en Cienzano, Bélgica, responsable del Laboratorio Nacional de Referencia para Campylobacter.
Hello everyone, my name is Koenraad van Hoorde. I am head of the Foodborne Pathogens Service at Cienzano, which is a public health institute in Belgium. Our lab is recognized as National Reference Laboratory by our Food Safety Agency, and this means that, amongst others, we are responsible for the detection, diagnosis, and surveillance of pathogens that can be transferred to humans via food. I was asked if I could talk today about MALDI-TOF MS, and it is with great pleasure that I will give some info on this technique and how it can be used in a context of food microbiology and food safety. There's a lot to say about this technology, so after a short introduction, I would like to tell a little bit more on how it works, how it can be applied to, and with a glance to future applications. So let's start with food and its microorganisms. I often like to refer to it as the good, the bad, and the ugly. On the one hand, you have the good ones. I think this speaks for itself. Those microorganisms we actually want to see in our food because they are important for flavor, texture, or because they enhance the nutritional quality. They can even have beneficial effects on our health. Then you have the bad ones, the spoilage microorganisms that reduce shelf life and make our food look and taste bad. Last but not least, we have the ugly ones, those that can constitute a health risk. It's obvious that a correct and unambiguous identification is crucial. So, to detect and identify these microorganisms in our food, we can use a variety of techniques, but the choice of technique depends on the actual goal and the information level required. Let's start from the food context. There are many reasons why we want to identify the microorganisms present in our food. Just because we want to know what is present and what they do, think about fermentations, or because we are looking for microorganisms with specific properties. We can also want to detect spoilers or pathogens, and this can then be put in a quality control perspective, or we can use it for source tracking. A given pathogen, where did it come from? Another important aspect is the level of information required. Methods vary in their resolution from family genus level up to strain characterization. To give an example of the importance of level of information, let's have a look at Salmonella. Taking into account the microbiological criteria for minced meat intended to be consumed raw, presence or absence of Salmonella is sufficient. This means that in none of the 5 samples of the sampling plan, Salmonella should be detected. For this, it is sufficient that, for example, with MALDI-TOF MS, suspected colonies are confirmed as being Salmonella. However, for outbreak investigation, it's important to characterize the strain in much more detail. We can do serotyping according to the Kaufman-White scheme, and we can even go much more in detail using whole genome sequencing, as was done during a recent multi-country outbreak investigation this summer in Europe. The third element is the state of the art. New methods arise, existing ones evolve. We can choose between phenotyping and genotyping methods, culture-dependent or independent approaches. We can even combine multiple methods in a polyphasic approach. All methods, they have their strengths and weaknesses, their advantages and disadvantages, on the level of time, ease of use, costs. But the most important, at the end, the method of choice should deliver the output you want to work with. Remember the context and the level of information. We are always looking for methods that are faster, better, with sufficient sensitivity, accuracy, and if possible, as cheap as possible. Well, for many applications, inevitably, you will bump onto MALDI-TOF MS. It offers high throughput, it has a high reproducibility, and the generated data, these are the mass spectra, they are relatively easy to work with. Well, for those that might not yet be familiar with the term MALDI-TOF-MS, it stands for Matrix-Assisted Laser Desorption Ionization Time-of-Flight Mass Spectrometry. The meaning will become clear in the following minutes. MALDI-TOF-MS, it's a mass spectrometry tool. So we are measuring the mass, it's better to say we are weighing molecules, and even more correctly, we are measuring the mass to charge ratio. Now, MALDI-TOF itself, it generates mostly singly charged ions, which means that in a spectrum, each molecule is represented by only one peak. Remember what I said about the fact that spectra are relatively simple. It is also a qualitative method. The presence of a peak means that a certain molecule is present, and sometimes the mass can provide info on the identity of the peak. In microbiology, for identification purposes in general, we will be measuring proteins, and we will be measuring between 2000 and 20,000 Dalton. The result is a characteristic fingerprint, but it is also possible to generate lipids or nucleic acids and turn these into fingerprints. Now, how does the technique work? Well, we start from a sample that is embedded in a matrix. The sample here is represented by the large blue, reddish, and white balls. The matrix is represented by the yellow balls. So this sample matrix mixture is targeted by a pulsed laser beam. And what happens is that the energy from the laser is transferred to the sample molecules via the matrix, and we get a sublimation. All analyte molecules are transferred directly into the gas phase. You can also see that during the same moment, the molecules or the analyte molecules are positively charged now. Now, in this process, the matrix plays a crucial role. It's thanks to the matrix that we are talking about the soft ionization. The matrix guarantees that high molecular weight molecules are not being fragmented. So we can actually analyze complete molecules and not fragments. Now, subsequently, a high voltage electric field is applied and the ionized charged molecules are accelerated towards the mass analyzer. All generated ions are in theory accelerated across the same distance by the same force. Hence, all ions have the same kinetic energy. Now, subsequently, in the time-of-flight tube, that's the mass analyzer, the ions are separated based on their mass over charge ratio. The larger the mass over charge ratio, the slower the ion will move. Now, of course, prior to the analysis, the sample needs to be prepared. There are different ways to prepare the sample. The most simple method, and this is the one that you can see on the left. So this method here, it's also the method that is most used in routine labs. We call it the direct smear method. It's sufficient to just with a loop or even a wooden toothpick to pick some cell material and to transfer it on the target plate. You should take care to not take too much cell material and to avoid to pick up some agar. Now, for microorganisms that are a little bit more difficult to lyse, sometimes additional steps need to be performed to obtain qualitative spectra. You can do this either by a targeted formic acid extraction. This enhances the release of proteins, or you can do a more elaborate ethanol formic acid extraction, which is in green indicated, and on the right you can see the different steps for this more elaborate protocol. Now, in each situation, at the end, sample material is applied on a target plate and the matrix is added. Now, when applying the matrix, you should try to obtain a nice homogenized analyte matrix sample, like is shown more at the bottom here. This way, you have a bigger success on a qualitative spectrum and you should try to obtain a thin layer. Important here is less is more. Of course, you also need a MALDI-TOF MS instrument, and different instruments exist, but again, they also have their advantages and disadvantages. On the left, you have the Microflex from Bruker. This will be replaced by a new model, the Sirius. In the middle, you have the Vitek MS from bioMérieux. Both the Microflex or Sirius and the Vitek MS, they come with own software and databases for identification. The device on the right requires the development of user-generated identification databases. On the other hand, it offers more possibilities in terms of analysis. Now, you have already, or you might have already seen that these instruments are quite high. This is because of the presence of the time of flight tube, so the mass analyzer, which can be up to 1.5 meters. And the mass analyzer is in this part of the instrument, so this large part here. Now, let's have a quick look at a movie of spectra acquisition. You can see repeated bright flashes here on the left. Each flash that you see corresponds with 200 shots of the laser. So it's actually very, very fast. You can also see that the spectrum is generated. And as already mentioned, the mass range is between 2000 and on the right, 20,000 Dalton. This is the range that is mostly used for identification purposes. During the analysis, you have a real-time identification going on. So we have here an identification that's going on, and in the meantime, the MALDI-TOF is continuing its analysis. And this goes on until all spots have been analyzed. Now, what is the result? A spectrum is generated. The next step is to find out what the spectral fingerprint is representing. Very briefly, the unknown profile is compared with a database of spectra of known origin. However, different manufacturers, think of Bruker and bioMérieux, they apply different ways to do this. However, also very important, the original spectra, they are pre-processed during the identification. Now, during that process, different things are going on. First, you can have a mass adjustment. So you can change the mass or the target mass range you want to analyze. Second, you can do a smoothing. And the smoothing is represented by the black towards blue curve. Smoothing results in a smoother curve. A third element is baseline subtraction. Baseline subtraction is represented by the green line here. So basically, you will see that where you had initially the black, then you had the blue, and the blue is really going down by the baseline subtraction. And last step is peak picking, where peaks of with specific characteristics can be selected. And so an actual raw mass spectrum is turned into a list of peaks. Now, this is a general principle. What happens now with that peak pattern? So on the left, you can see that a peak list of peaks of the unknown sample, which is on the top, is compared with a peak list of a known spectrum. And for identification, an algorithm is looking at first which peaks are present in both in the unknown and in the reference pattern. And then also the algorithm will look at which peaks are present in the unknown, which are the red ones, but not present in the reference pattern, and also vice versa, which peaks here in blue are present in the reference spectra, but not present in the unknown spectrum. And a score is calculated, and this is turned into a log score. And depending on the value of the log score, the interpretation is different. So when you have a log score between 2 and 3, you will have a high confidence identification. Between 1.7 and 1.99, you have a low confidence identification, and below that you have no identification possible. Now there's also a second parameter which refers to the consistency of the identification. And to give you an idea about that, so we have two examples here. On the top here, you have a high confidence and also high consistency identification of, for example, a Campylobacter jejuni. On the bottom here, you have, despite the fact that you have a high confidence identification, you have only a low consistency identification. This because you have two different species that have been identified, but it makes the interpretation rather difficult because which of those species is the actual unknown that was being analyzed. So this is also something to take into account. Now, how do we do it here at Cienzano, or how are we using MALDI-TOF? So we mainly use MALDI-TOF as a confirmation step. For example, for strains of Salmonella, Listeria monocytogenes, E. coli, Clostridium perfringens, Yersinia enterocolitica, Vibrio parahemolyticus, Cholera and Vunificus, Conobacter and Campylobacter species. To be able to do this, we performed also an in-house validation, including inclusivity, exclusivity. We participated in various proficiency tests in which we, in parallel, analyzed the unknown isolates using API tests. And based on that, since 2016, we were ISO 17025 accredited by our national accreditation body. For identification, we are not accredited, so it's merely for confirmation purposes. Now, what do we have? So in our library, we have the MALDI Biotyper research use only database, which contains almost 8,000 reference spectra. We also have the SR database. We use the Listeria subtyping module. We have the MALDI Biotyper Mycobacterium and filamentous fungi databases. Now, regarding the use of MALDI-TOF at our lab, so what are the positive aspects? So first of all, in most cases, we use the direct smear method. So it's really a very simple protocol, which means that regarding operating costs, they are very low. Regarding consumables, also staff, you generate limited waste, and the results, you obtain really fast results and can immediately be interpreted. Remember the short movie that I showed you? During the analysis, you already get identification. And also a good thing is that the existing databases can be expanded. The disadvantages of the technique, the purchase of the instrument and also maintenance are very expensive. There are some limits of identification. Remember also the example that I showed. And sometimes also additional information that you would be interested in is very limited. Now, this brings me to some other future applications. I would not say really future because these things are really ongoing, but maybe rather future towards really routine labs. So it's more research applications for the moment, but hopefully maybe towards the future, this will also be able to be implemented in routine labs. So a first element is the use of MALDI-TOF MS as a rapid tool for the detection of antimicrobial resistance. This can be done by three approaches, or just by looking at the characteristic difference in the profile between a sensitive and a resistant strain. So you could look at specific peaks, which are called biomarkers. This is on the top left, an example. Or you could do it via a more functional approach, like monitoring the cleavage of an antibiotic by an antibiotic-resistant strain. Here in the example, you can see that a beta-lactam antibiotic is cleaved by a beta-lactamase producing bacterium. So the colored peaks, you get a shift in the mass because of the degradation of the antibiotic. And then on the right, you have like a process that is similar to minimal inhibitory concentration tests, but here you are going to look at the minimal concentration at which the mass spectra will change. And this is here somewhere near 0.5 micrograms per milliliter. Now, another application is the use of MALDI-TOF MS as a tool for typing. Here, an example of Klebsiella pneumonia. Different phylogroups can be differentiated one from another, indicated by the specific boxed peaks here. And then a last application, MALDI-TOF can also, or there are possibilities to use MALDI-TOF as a culture-independent tool, just to show and to end. We were able to make a difference between extractions from fermented milk and the corresponding kefir grains. And then we were also able to assign some biomarker peaks that are characteristic for Lactobacillus kefiranofaciens, which we could only find in the profiles from the kefir grains indicated here in yellow. Okay, so this was the end of my presentation. I hope that I was able to give you some information about what MALDI-TOF is and how it can be used in a routine lab. And I hope also the info on future applications was helpful. Thank you.